Hello everyone. If you don't remember me from my last presentation, again, my name is Alejandro Ayala. Welcome back and welcome to my presentation on the regulation of glycolysis, gluconeogenesis, and the enzyme aldolase that is involved in the process. Okay, we're going to start off with an overview. Today, we will be talking about glycolysis, gluconeogenesis, and aldolase. We are going to start off with an overview of the lesson and what I will present. That's what we're doing now. Before we dive in, we will take a step back and look at the big picture. I will briefly explain glycolysis, gluconeogenesis, and the enzyme aldolase. We will then get into the subject of glycolysis. I will give an overview of the glycolysis pathways, how it is regulated, and the role of aldolase in the glycolysis pathway. We will then get into we'll then discuss gluconeogenesis. Like I did with glycolysis, I will give an overview of the gluconeogenesis pathways, how it is regulated, and the role of aldolase in gluconeogenesis. We will then shift our focus to the enzyme aldolase. I will explain the different isoforms of aldolase and their different expression patterns. We will examine how aldolase B expression can be regulated by transcription factors HNF1, HNF3, CEPB, and DBP. I will conclude my presentation with one example of current research related to this topic. Okay. So, what is the big picture? Glycolysis is the breakdown of sugar for energy in the usable form for our bodies. Glycolysis, as we know from other courses, is dealing with sugars, and lysis is the breaking apart of. Gluconeogenesis is the synthesis of glucose from organic but non-carbohydrate material. That is, there's something in our body, there's organic materials in our body that are not carbohydrate sugars. There are other things, and they get put together to make glucose. Aldolase is a protein that helps break down certain sugars for energy. In our bodies, we possess the machinery needed to convert food into energy that our cells can utilize to carry out life-sustaining functions. Glycolysis is the process by which our body's machinery converts food to energy. So glycolysis is turning food into energy. Because of the continual need for energy to fuel those functions, it is necessary that our bodies have access to its food source. This is what gluconeogenesis is for, to supply a continual source of glucose for our cells. Understanding how the entire process is regulated and how those regulators work can help develop healthy behavioral strategies, as well as develop drugs that have a pharmacological effect in the tissues and proteins involved. Okay, now we're going to go into glycolysis. What is glycolysis? This is an overview of the pathway. ATP is often referred to as the energy currency of cells. Glycolysis produces ATP as well as NADH and pyruvate, which are materials that will later be used to make more ATP in other metabolic pathways that we won't cover in this lesson. So glycolysis does produce ATP, but in short amounts, as well as NADH in short amounts. But the most important part there is that it produces pyruvate, which goes into the other metabolic pathways that actually yields a lot more ATP. Okay, glycolysis occurs in the cytoplasm of the mitochondria and can occur under aerobic or anaerobic conditions. Aerobic conditions result in pyruvate synthesis, which diffuses into the mitochondrial matrix for other metabolic pathways. Under anaerobic conditions, pyruvate remains in the cytosol to be used as a reducing agent to regenerate NAD+. And this keeps glucose flowing into the glycolytic pathway. So even though there's no oxygen, because of what pyruvate turns into, it'll allow glucose to be getting fed into the system, and that does produce two molecules of ATP, which is really low, but it's something. Okay, now, depending on the source that you're reading, glycolysis is broken down into either 2 or 3 phases. The 2-phase model divides glycolysis into the preparatory phase, in which glucose is phosphorylated and converted to glyceraldehyde 3-phosphate. The second phase of this 2-phase model is the payoff phase, which consists of the oxidative conversion of glyceraldehyde 3-phosphate being oxidized and converted to pyruvate along with the products NADH and ATP. In the 3-phase model, the only difference is that the cleaving of fructose 1,6-biphosphate into 2 molecules of a 3-carbon unit glyceraldehyde 3-phosphate. The only difference is that this cleaving is categorized separately into its own phase, and that's right smack in the middle. This is the second phase that is created when it's in a 3-phase model. But a lot of resources like the text by Nelson and Cox 2017, it lists it as a 2-phase model. Now, this process involves several steps, enzymes, and products that actually feed back into the loop, and it'll either increase or decrease the products made. So it's a really complicated system, as you well know. But the main idea is that aerobic glycolysis results in 2 ATP, 2 NADH, and 2 pyruvate molecules. These aren't very much, but remember, the glycolysis step feeds the later metabolic pathway steps that actually produces the greater amounts of ATP. Okay. What is glycolysis? How it is regulated. That's what we're going to talk about now. When food is eaten, it is broken down into glucose and released in the blood, where it binds to glucose receptors. Upon binding of glucose, insulin is then released and eventually binds to its receptor. That results in a transduction signal that translocates GLUT4 transport protein into the plasma membrane. Once inside the cell, glucose is in the presence of the hexokinase enzyme that will phosphorylate glucose into glucose 6-phosphate, which is the first step in glycolysis. This enzyme undergoes competitive inhibition in great amounts of its product, glucose 6-phosphate. So this hexokinase, it gets a negative, it gets a feedback inhibition from its own product. So once it starts making a lot more product, it'll eventually stop, because that product will just by sheer number inhibit it from attaching to the other molecules and producing more of the product. Now, this is kind of complex and difficult to understand, so I'm going to go ahead and just go over it again. So we eat something. Let's say it's a cookie. And that's, as you can see in the image here, the cookie would be the glucose that we take into our system. Now, the top portion is the blood flowing back and forth. So we have glucose in our blood. So once we have glucose in our blood, the glucose binds to glucose receptors. Now, once that happens, insulin is released by the pancreas, I believe. Now, insulin is released and it goes to the insulin receptors. What I meant by transduction signaling is that insulin attaches at the extracellular side, and it creates a signal that goes down to that vesicle that we see right here. Now, this vesicle, it has these we can think of them as channels that are connected to the vesicle. Now, once the insulin activates this protein, and then this activates this, the vesicle will release this channel, so glucose can now flow inside. Remember, in here, there are hexokinase enzymes. So once glucose comes in contact with hexokinase, that right there is going to be the first step of glycolysis. All right, next one. What is glycolysis and the role of aldolase in it? Aldolase is the glycolytic enzyme that um, aldolase is the glycolytic enzyme that catalyzes the breakdown of fructose 1,6-diphosphate to glyceraldehyde 3-phosphate. Aldolase takes up the open-ringed form of fructose 1,6-biphosphate made possible by a shift-based reaction. So, aldolase is an enzyme, and fructose comes in as a ring structure. We know sugars are ring structures. Fructose, it does not want to do this, but there's like a 1 to 2% occurrence of the fructose getting split into a open ring structure. Now, once this open ring structure occurs, it can actually slide into the aldolase enzyme, and then that's how the different functional groups of the aldolase enzyme are able to interact with that fructose. So which functional groups are we talking about? Lysine 223, arginine 295, and glutamic acid 178 are in the aldolase and are the key functional groups that cleave fructose into two molecules. The cleavage of fructose releases oxygen in the form of water, and it also involves breaking and creating of double bonds between carbon-nitrogen, carbon-oxygen, and nitrogen-oxygen. Now, this occurs because the lysine, arginine, and glutamic acid are in different spatial areas. So these mechanisms they happen in different steps, and one can't happen before the others happen. So it's a stepwise process. And if you look at the references that I have on my presentation, there's the images and resources there so you can get a better idea of what this actually looks like. And we'll have an image here coming up that's going to show a little bit of that as well. The point here is that aldolase cleaves fructose into two molecules. Now, these two molecules is what actually allows for two pyruvates to be made. Here, I must add that one of the products of aldolase requires one additional step catalyzed by triose phosphate isomerase to create the second molecule of glyceraldehyde 3-phosphate, which is what actually continues down the glycolytic pathway. I'll repeat that. So, aldolase cleaves the fructose into two molecules. One of those molecules needs to get converted to the glyceraldehyde 3-phosphate because that is the one that's going to continue on in this glycolysis process. Okay, and the video is covering it, but here I have on the screen a picture of the molecule of glyceraldehyde 3-phosphate. This is what's going to be going down into the lower levels of the glycolytic pathway, and this other molecule here is what actually gets converted into, so this is going to get converted into this. Okay. Now we're going to go into gluconeogenesis. What is gluconeogenesis? We're going to start off with an overview of the pathways. Glucose can be found in the blood or stored as glycogen to be made to be used to make ATP for energy. So glucose is either in the blood or stored as glycogen, and this is then used to make ATP. Now, sometimes the amounts of glucose in the blood and the glycogen stores that we have is not enough. Whether you're fasting, whether you're doing strenuous exercise, where you're using up all the glucose that's available in your system, that's readily used to make ATP, it's just not going to be enough, and we're going to need a process to make glucose from the materials in our system. Now, gluconeogenesis means new formation of sugar, and this process is found in plants, animals, and fungi. This is a process that's has been found across time and across taxa. So this is very important for life and for energy and physiological processes. Now, In mammals, this process occurs mostly in the liver, kidneys, and lining of the small intestine. Okay. The first, third, and the last step of glycolysis greatly favors the products. So this means that glycolysis has 3 steps, the first, third, and last, that are really favored towards the product side. That means that glycolysis wants to occur. Glycolysis wants to turn that glucose into the products, pyruvate and NADH and ATP. So what do I mean by it's favored towards the products? The total Gibbs free energy for glycolysis is -84 kilojoules per mole, and that makes it a spontaneous reaction. We know that anything that has a negative Gibbs free energy is going to happen spontaneously. In order for gluconeogenesis to occur, glucose 6-phosphatase reverses step 1. Fructose 1,6-biphosphatase reverses the third reaction, and pyruvate carboxylase reverses the last reaction. Okay, so we have those 3 really negative Gibbs-free reactions. So those are sort of like barriers that we have to overcome. There are 3 enzymes that help us do that, and that's what I just listed: the fructose biphosphatate, the pyruvate carboxylase, and the glucose 6-phosphatase. So because of those 3 enzymes, gluconeogenesis can proceed, despite the the trend towards the product side of glycolysis. All right. What is gluconeogenesis and how is it regulated? I'm sorry. Okay. So glucagon, growth hormone, epinephrine, and cortisol are termed diabetogenic hormones that stimulate gluconeogenesis in the kidneys and liver. Cortisol, for example, stimulates gluconeogenesis by decreasing glycogen synthesis. They cause the muscle cells to decrease glucose uptake, and it indirectly causes protein degradation. Increased lipolysis occurs in the adipose tissues in the fat cells. Cortisol decreases insulin. Cortisol also increases glucagon. Now, all those things, what they do is they cause the body to trend towards making glucose from the materials that you have in your system. So you're not breaking down glucose. In a sense, let's just say that you have too much, let's say that you don't have enough glucose in your system, so you're wanting to make glucose from the materials that you do have. Okay. What is gluconeogenesis and the role of aldolase? Aldolase is the enzyme that converts 2 molecules of 1,3-biphosphoglycerate into 2 molecules of glyceraldehyde 3-phosphate in a reaction which oxidizes NADH and releases inorganic phosphate. This reaction is possible because under physiological conditions, delta G is only -6. Here, I must say that this is important to remember. We are talking about synthesis of glucose and not the other way around. So going in the direction of gluconeogenesis, the synthesis of glucose, the 2 reactions that come before the aldolase favor the products, which in effect favors the following reaction products. And so this is sort of like a cascade effect because of principles of equilibrium. If your precursor is in high amounts, the following reaction is going to be trending towards the products as well, because you're going to have a lot of reagents and you're going to use those reagents to make products, which you don't have much of. So it's sort of like a cascading effect. That's what drives it towards the development of glucose. The delta G's in the previous reactions are anywhere between 0 to -4 for one reaction and between -2 and 2 for the other reaction. Okay, I'm going to explain that a little bit. So we have two sugars that we're going to use aldolase for to make the 1-fructose molecule. Now, those those delta G's, the Gibbs-free energies, they are negative. So to go from the 2 separate sugars to the 1-fructose, that right there is going to be that actually is the -6 Gibbs free energy, but it's the precursor to that. That's actually what I have listed here as a -4, and the range from -2 to 2. So it's not necessarily the step from the 2 sugars to the 1-fructose. It's the one previous to that sort of helps drive the reaction forward to the formation of glucose. So if we look at my images here, this is sort of what I'm talking about down here. So we have these 2 separate molecules, and aldolase, if you go towards glucose, you're going to be pushing this reaction this way. Okay. Now we're going to be talking about aldolase. Aldolase and isoforms. Aldolase A is the isoform associated mostly with glycolysis and the cleavage of fructose glycerol. Mutations to this gene are relatively mild, with one example being non-spherocytic hemolytic anemia. Though the known mutations to this isoform are mild, because it is the isoform found in mammalian embryos, it could mean that any mutation to this enzyme substantially increases the chances of death, which is why more serious mutations are not found. That means mutations in aldolase A are mostly seen as mild, but that's because any more serious mutations automatically results in death. So that's what's suspected there. So it's really, if you have a mutation that's, relatively significant, it's really likely that it's actually causing embryonic death there. Okay. Let's see. Aldose B. This is mostly known for fructose metabolism in gluconeogenesis. So aldolase B is part of gluconeogenesis mostly, and it is found mostly in the liver. Mutations to this gene cause fructose intolerance. Those who have a high diet of fructose, they suffer because they can't metabolize the fructose because they don't have that aldolase B enzyme that helps metabolize that. Okay, this isoform has been shown to have up to a 30-fold greater efficiency in fructose metabolism than the other enzymes. So that makes it more so associated with the gluconeogenesis. Similarities between isoenzymes include physical, chemical, and active sites, as well as the main functional group residues being placed on the outside of the protein. So the isoenzymes are different and they're located in different places, but they have similar chemical properties. Okay. Expression patterns. All aldolase isoforms come from a single ancestral gene, like I was talking about earlier. It is known to have occurred in different taxa throughout time. Aldo A is the primary isoform from mammalian embryos and is more concentrated in muscle tissue. Aldo B is the primary isoform in bird and amphibian embryos, and it is expressed mostly in the liver, kidneys, and lining of the small intestine. Again, that one's associated with gluconeogenesis. Aldo C is found mostly in CNS tissue, the brain, neurons, and those kinds of things that have smooth muscle as well. Okay. Now, talking about aldolase transcription factors. Transcription, of course, is actually making aldolase enzyme. We are now going to be talking about transcription factors HNF1 and HNF3. Now there's two terms I wanted to define here is activator and transactivator. Activators are proteins that help initiate gene transcription of genetic code, and transactivators are proteins that speed up the rate of transcription. Transcription factor HNF1 regulates ALDO-B by being a strong transactivator, and this means that it increases the rate of gene transcription for ALDO-B. Transcription factor NHF3 is a very strong competitive antagonist to HNF1, though it does not do so by itself. It does nothing to the promoter for ALDO-B. It is also a non-competitive antagonist to DPP, and I'm going to talk about what DPP does here in a sec. So first we'll discuss CEPB. Transcription factor CEPB regulates ALDO-B expression by being an activating binding site to the ALDO-B promoter, though it is categorized as a weak activator. The transactivation by this factor increases by deleting a distal negative element. And a negative element is something downstream that causes stops or slows down or has a negative effect on transcription. In liver cells, this factor is a strong transactivator. Transcription factor DPB regulates ALDO-B expression by being an activating binding site to the ALDO-B promoter as well. This factor is also a strong transactivator. This factor transactivates ALDO-B transcription to a lesser extent when a distal negative element is removed. In liver cells, this gene is not a factor. So it's a lot of information that was just presented to you, but the idea is that these four transcription factors, they impact the transcription of ALDO-B, and just by going over which transcription factor does what, you'll know if it's helping it start up the transcription, if it's helping it increase the rate at which is being transcribed, or if it's stopping it, and how it's stopping it, of course, right? If it's competitive or non-competitive, antagonist, that kind of thing. All right, so we're here to our last point, and it's about current research being done related to this field. As mentioned previously in this presentation, ALDO-B is associated with fructose metabolism difficulties. One recent study involved looking at asymptomatic carriers for hereditary fructose intolerance found that having the gene, even though it's recessive, increased susceptibility to the negative impacts of a high fructose diet, and this affects uric acid levels and hepatic insulin sensitivity. That is to say, only the heterozygotes showed an increase in uric acid levels and liver insulin resistivity immediately following a meal high in fructose. So basically, the people who are carriers of a mutation in the ALDO-B gene, the people who have the mutation there, they are not able to balance out their chemicals. Their uric acid levels go up and their liver insulin counts are high. That means that they are desensitized to it, so they need to have more insulin. The authors do caution that acute ATP deficiency occurred in both the experimental and control groups. ATP deficiency occurred in both groups, and that wasn't controlled for. It wasn't clearly stated, okay, ATP deficiency doesn't cause this or it doesn't cause that. So further research is going to involve looking at how ATP deficiency comes into play, and also looking more at metabolic disorders that also raise uric acid levels to do comparisons. Okay. In my image here, I have a picture of gout, which is caused by increased levels of uric acid. Now, we are into our last slide, the conclusion. So today we spoke about glycolysis, gluconeogenesis, and aldolase. I gave an overview of glycolysis pathways, how it is regulated, and the role of aldolase. I did the same for gluconeogenesis. We then shifted to the enzyme aldolase. I explained the different isoforms of aldolase and the different expression patterns, how they showed up in different tissues and things like that, and what they did, whether it was for smooth muscle contraction. We know that Aldolase B, of course, was dealing with fructose metabolism, and so anyways, moving on. We examined how aldolase B expression can be regulated by transcription factors HF1, HNF1, HNF3, CEPB, and DPB. I concluded my presentation with an example on current research related to this topic. Glycolysis and gluconeogenesis has been evolutionarily preserved across time. By growing our knowledge of these pathways, the enzymes, and the transcription factors involved, we can be confident that healthcare advances in the specific science will yield great benefit. Something so inherently tied to our physiology must be explored and understood. Thank you very much.